So, continuing with the chapter Mechanism of Action of Enzymes, comes the topic chymotrypsin and fructose 2,6 bisphosphate illustrate covalent catalysis. Chymotrypsin. While catalysis by aspartic proteases involves the direct hydrolytic attack of water on a peptide bond, catalysis by serine protease chymotrypsin involves formation of a covalent acyl enzyme intermediate. A conserved sterile residue, serine 195 in the bovine form, is activated by interaction with histidine 57 and aspartate 102. While three residues are far apart in primary structure, in the active site of the mature folded protein, they reside within bond forming distance of one another, aligned in the order as 102 his 57 ser 195. This trio forms a linked charge relay network that acts as a proton shuttle. Binding of substrate initiates proton shifts as that in effect transfer the hydroxyl protons of serine 195 to aspartate 102. See figure 77. You can see um, this is the catalysis by chymotrypsin. In the first diagram, we can see that the charge relay system this removes a proton from serine 195, making it stronger nucleophile. Secondly, we can see that activated ser 195 attacks the peptide bond, forming a transient tetrahedral intermediate. In the third figure, it is shown that the release of amino terminal peptide is facilitated by donation of a proton to the newly formed amino group by his 57 of the charge relay system, yielding an acyl ser 195 intermediate. Fourth, his 57 and ASP102 collaborate to activate a water molecule which attacks the acyl 195, forming a second tetrahedral intermediate. In the fifth figure, we can see that the charge relay system donates a proton to cell 195, facilitating the breakdown of the tetrahedral intermediate to release the carboxy terminal peptide, as in figure 6. Okay, the enhanced nucleophilicity of the sterile oxygen facilitated attack on the carbon of the peptide bond of the substrate, forming a covalent acyl enzyme intermediate. The proton on ASP102 then shuttles via his 57 to the amino group liberated when the peptide bond is cleaved. The portion of the original peptide with a free amino group then leaves the active site and is replaced by a water molecule. The charge relay network now activates the water molecule by withdrawing a proton through his 57 to ASP102. The resulting hydroxide ion joins the acyl enzyme intermediate and a reverse proton shuttle returns a proton to cell 195, restoring its original state. While modified during the process of catalysis, chymotrypsin emerges unchanged on completion of the reaction. The proteases trypsin and lastase employ a similar catalytic mechanism, but the numbering of the residues in their cis-hip as proton shuttles differ. Next comes fructose 2,6 bisphosphatase. Fructose 2,6 bisphosphatase, a regulatory enzyme of gluconeogenesis, catalyzes the hydrolytic release of the phosphate on carbon 2 of fructose 2,6 bisphosphate. Figure 7.8 illustrates the role of 7 active site residues. Catalysis involves catalytic triad consisting of one glue and two his residues, of which one his forms a covalent phosphodiestidyl intermediate. As we can see in figure 7.8, this is the catalysis by fructose 2,6 bisphosphate. In first, we can see lies 365 and R's 257, 307 and 352 stabilize the quadruple negative charge of the substrate by charge-charge interaction. Glue 327 stabilizes the positive charge on his 392. In the second figure, we can see that the nucleophile his 392 attacks the C2 phosphoryl group and then transfers it to his 258, from forming a phosphoryl enzyme intermediate. Fructose 6 phosphate now leaves the enzyme. In the third figure, we can see the nucleophilic attack by a water molecule, possibly assisted by glue 327, acts as a base, forms inorganic phosphate. In the fourth figure, we can see inorganic orthophosphate is released from arginine 257 and arginine 307, reproduced with permission from some writer. Okay. So now coming back, this illustrates the role of 7 active site residues and this can see the catalytic triad. Next topic is catalytic residue are highly conserved. Members of an family, such as the aspartic or serine proteases, employ a similar mechanism to catalyze a more common reaction type but act on different substrates. Most enzyme families appear to have arisen through gene duplication events that created a second copy of the gene that encoded a particular enzyme. The two genes and consequently the encoded protein can then evolve independently, forming divergent homologues that recognize different substrates. The result is illustrated by chymotrypsin, which cleaves peptide bonds on the carboxy terminal side of large hydrophobic amino acids, and trypsin, which cleaves peptide bonds on the carboxyl terminal sides of basic amino acids. Proteins that diverge from common ancestors are said to be homologues to one another. The common ancestry of enzymes can be inferred from the presence of specific amino acids in the same relative position in each family member. These residues are said to be evolutionarily conserved. Isoenzymes are distinct enzyme forms that catalyze the same reaction. Higher organisms often elaborate several physically distinct versions of a given enzyme, each of which catalyzes the same reaction. These protein catalysts or isoenzymes can arise through gene duplication or in high eukaryotes alternative mRNA splicing. While the homologous proteases described act on different substrates, isoenzymes also may differ in auxiliary features such as sensitivity to a particular regulatory factors or subcellular location that adapt them to specific tissues or circumstances rather than distinct substrates. Isoenzymes that catalyze the identical reaction may also enhance serving by providing a backup copy of essential enzyme. The catalytic activity of the enzymes facilitates their detection. The relatively small quantities of enzymes typically contained in cells hamper determination of their presence and abundance. However, the ability to rapidly transform thousands of molecules of a specific substrate into products imbues each enzyme with the ability to simplify its presence under appropriate conditions. The rate of the catalytic reaction being monitored is proportionate to the amount of the enzyme present, which allows its concentration to be inferred. Assays of the catalytic activity of enzymes are frequently used both in research and clinical laboratories. Single molecule enzymology. The limited sensitivity of traditional enzyme assays necessitates the use of a large group or ensemble of enzyme molecules in order to produce measurable quantities of the product. The data obtained thus reflects the average activity of the individual enzymes across multiple cycles of catalysis. Recent advances in nanotechnology and imaging have made it possible to observe catalytic events involving discrete enzyme and substrate molecules. Consequently, scientists can now measure the rate of the individual catalytic events and sometimes a specific step in catalysis by a process called single molecule enzymology, an example of which is illustrated in figure 7.9. So in this figure, we can see the direct observation of single DNA cleavage events catalyzed by restriction endonuclease. DNA molecules immobilized to beads blue are placed in the flowing stream of buffer black arrows, which causes them to assume an extended conformation. Cleavage at one of the restriction site orange by an endonuclease leads to the shortening of the DNA molecule, which can be observed directly in a microscope since the nucleotide bases in DNA are fluorescent. Although the endonuclease red does not fluoresce, hence it is invisible, the progressive manner in which DNA molecule is shortened 1 to 4 reveals that the endonuclease binds to the free end of the DNA molecule and moves along it to form site to site. Drug discovery requires enzyme assays suitable for high throughput screening. Enzymes are frequent targets for the development of drugs and other therapeutic agents. These generally take the form of enzyme inhibitors. The discovery of new drugs is greatly facilitated when a large number of potential pharmacophores can be simultaneously assayed in a rapid automated fashion, a process referred to as high throughput screening HTS. HTS employs robotics, optics, data processing, and microfluidics to simultaneously conduct and monitor thousands of parallel enzyme assays. It thus serves as perfect complement to combinatorial chemistry, a method for generating large libraries of chemical compounds spanning all possible combinations of a given set of chemical precursors. Enzyme linked amino acids. The sensitivity of enzyme assays can be exploited to detect proteins that lack catalytic activity. Enzyme-linked immunosorbent assays (ELISAs) use antibody covalently linked to a reporter enzyme such as alkaline phosphatase or horseradish peroxidase that can be used to produce easily detected chromogenic or fluorescent products in vitro. Serum or other biologic samples to be tested are first placed in multiple microtiter plates. Most proteins adhere to the plastic surface and are thus immobilized. Any exposed plastic that remains is subsequently blocked by adding a non-antigenic protein such as bovine serum albumin. A solution of antibody covalently linked to a reporter enzyme is then added and the antibody is allowed to adhere to any immobilized antigen molecules. Excess free antibody molecules are then removed by washing. The presence and quantity of bound antibody is then determined by assaying the activity of the reporter enzyme, a quantity that should be proportional to the number of antibody molecules present and presumably the antigen molecules to which they are bound. NADP plus dependent dehydrogenases are assayed spectrophotometrically. The physiochemical properties of the reactants in an enzyme-related reaction dictate the options for the assay of enzyme activity. Spectrophotometric assays exploit the ability of a substrate or product to absorb light. The reduced coenzymes NADH and NADPH, written as NAD(PH), absorb light at wavelength 340 nm, whereas their oxidized forms NADP plus do not. When NADP plus is reduced, the absorbance are at 340 nm therefore increases in proportion to and at a rate determined by the quantity of NADPH. So in the diagram we can see a peak at 340. Okay. Absorption spectra of NAD plus and NADH densities are for 44 mg per liter solution in a cell with 1 cm light. But the NADP plus and NADPH have spectra analogous to NAD plus and NADH respectively. Now, the NADH produced. Conversely, when dehydrogenase catalyzes the oxidation of NADPH, a decrease in absorbance at 340 nm will be observed. In each case, the rate of change in absorbance at 340 nm will be proportionate to the quantity of the enzyme present. The assay of enzymes whose reactions are not accompanied by a change in the absorbance or fluorescence is generally more difficult. In some instances, either the product or remaining substrate can be transformed into a more readily detected compound. Although the reaction produced may have to be separated from unreacted substrate prior to the measurement, an alternative strategy is to devise a synthetic substrate whose products absorb light or fluoresces. For example, hydrolysis of the phosphoester bond in paranitrophenyl phosphate PNPP, an artificial substrate molecule is catalyzed at a measurable rate by numerous phosphatases, phosphodiesterases, and serine proteases. While PNPP does not absorb visible light, the anionic form of the paranitrophenol pK 6.7 generated on its hydrolysis strongly absorbs light at 419 nm and thus can be quantified. Many enzymes may be assayed by coupling to a dehydrogenase. Another quite general approach is to employ a coupled assays, figure 711. Typically, a dehydrogenase whose substrate is the product of the enzyme of interest is added in catalytic excess. The rate of appearance or disappearance of NADPH then depends on the rate of enzyme reaction to which the dehydrogenase has been coupled. The analysis of certain enzymes aids diagnosis. So in this figure 711 we can see the glucose is converted to glucose 6-phosphate, which is converted to 6-phosphogluconolactone. Now the production of glucose 6-phosphate by hexokinase is coupled to the oxidation of this product by glucose 6-phosphate dehydrogenase in the presence of added enzyme NADP+. When excess of the glucose 6-phosphate dehydrogenase is present, rate of formation of NADPH, which can be measured at 340 nm, is governed by the rate of formation of glucose 6-phosphate by hexokinase. Now, the analysis of enzymes in blood plasma plays a central role in the diagnosis of several disease processes. Many enzymes are functional constituents of blood. Examples include pseudocholinesterase, lipoprotein lipase, and components of the cascades that trigger blood clotting, clot dissolution, and opsonization of invading microbes. Several enzymes are released into plasma following cell death or injury. While these latter enzymes perform no physiologic function in plasma, they can serve as biomarkers, molecules whose appearance or levels can assist the diagnosis and prognosis of diseases and injuries affecting specific tissues. The plasma concentration of an enzyme or other protein released consequent to injury may rise early or late and may decline rapidly or slowly. Cytoplasmic proteins tend to appear more rapidly than those from subcellular organelles. Quantitative analysis of the activity of released enzymes or other proteins, typically in plasma or serum but also in urine or various cells, provide information concerning diagnosis, prognosis, and response to treatment. Assays of enzyme activity by typically employ standard kinetic assays of initial reaction rates. Table 7.1 lists the several enzymes of value in clinical diagnosis. Note that these enzymes are not absolutely specific for the indicated disease. For example, elevated blood levels of prostatic acid phosphatase are associated typically with prostate cancer, but may also occur with certain other cancers and non-cancerous conditions. Interpretation of enzyme assay data must take due allowance for the sensitivity and diagnostic specificity of the enzyme test, together with other factors elicited through a comprehensive clinical examination that includes patient's age, sex, prior history, and possible drug use. Analysis of serum enzymes following tissue injury. An enzyme useful for diagnostic enzymology should be relatively specific for the tissue or organ under study and should appear in the plasma or other fluid at this time useful for diagnosis the diagnostic window. In case of myocardial infarction MI, detection must be possible within a few hours or less of a preliminary diagnosis to permit initiation of appropriate therapy. The first enzymes used to diagnose MI were aspartate aminotransferase AST, alanine aminotransferase ALT, and lactate dehydrogenase LDH. Diagnosis using LDH exploits the tissue specific variations in its quaternary structure figure 712. However, it is released relatively slowly from following injury. Creatine kinase CK has three specific isoenzymes CKMM skeletal muscle, CKBB brain, and CKMB heart and skeletal muscle, along with a more optimal diagnostic window. As with LDH, individual CK enzymes isoenzymes are separable by electrophoresis. So in this figure you can see normal and pathologic patterns of lactate dehydrogenase isoenzyme in human serum. Samples of serum were separated by electrophoresis. LDH isoenzymes were then visualized using a dye-coupled reaction for specific for LDH. Pattern A is serum from a patient with a myocardial infarct. B is normal serum, and C is serum from a patient with liver disease. Arabic numerals identify LDH isoenzymes 1 through 5. Electrophoresis and specific detection technique thus can be used to visualize isoenzymes of enzymes other than LDH. In this table, we can see the serum enzyme, which is like alanine aminotransferase is major diagnostically used in viral hepatitis, amylase in acute pancreatitis, ceruloplasmin in hepatolenticular degeneration, Wilson disease. Creatine kinase in muscle disorders, gamma glutamyl transferase in various liver diseases, lactate dehydrogenase isoenzyme 5 in liver diseases, lipase in acute pancreatitis, beta cerebroside in Gaucher disease, phosphatase acid in prostatic disease including cancer, phosphatase alkaline isoenzymes various bone disorder, obstructive liver diseases. Okay. Now, coming back to it, today the assays of plasma CK levels are primarily used to assess skeletal muscle disorders such as Duchenne muscular dystrophy. Plasma troponin constitutes the currently preferred diagnostic marker. Troponin is a complex of three proteins present in the contractile apparatus of skeletal and cardiac muscle but not in smooth muscle. Troponin levels in plasma typically rise for 2 to 6 hours after an MI and remain elevated for 4 to 10 days. Immunological measurements of plasma levels of cardiac troponins I and T thus provide sensitive and specific indicators of damage to heart muscle. Since other sources of heart muscle damage also elevate serum troponin levels, cardiac troponins provide a general marker of cardiac injury. Additional clinical use of enzymes. Enzymes are employed in the clinical laboratory to determine the presence and the concentration of critical metabolites. For example, glucose oxidase frequently is utilized to measure plasma glucose concentration. Enzymes also are employed with increasing frequency for the treatment of injury and disease. Examples include tissue plasminogen activator TPA or streptokinase for treatment of acute MI and trypsin for treatment of cystic fibrosis. Intravenous infusion of recombinantly produced glycosylases can be used to treat lysosomal storage syndromes such as Gaucher disease, beta glucosidase, Pompe disease, alpha glucosidase, Fabry disease, alpha galactosidase A, Sly disease, beta glucuronidase, and mucopolysaccharidosis types 1, 2, and 6, also known as Hurler syndrome, alpha L iduronidase, Hunter syndrome, iduronate 2 sulfatase, and Mantrollex-Lamy syndrome, aryl sulfatase B, respectively. Enzymes facilitate diagnosis of genetic and infectious diseases. Many diagnostic diseases take advantage of the specificity and efficiency of the enzymes. Many diagnostic techniques take advantage of the specificity and efficiency of the enzymes that act on oligonucleotides such as DNA. Early on, restriction endonuclease emerged as an important tool for both clinical and forensic analysis. Restriction endonucleases, often referred to as restriction enzymes, cleave the double-stranded DNA at sites specified by a consequence of 4, 6, or more base pair called restriction sites. If an individual bears a mutation or polymorphism that eliminates or generates the restriction site for one of the endonuclease being used, the number and size of DNA fragments produced on cleavage will differ from the other individual. Such restriction fragment length polymorphism RFLPs can be used for prenatal detection of deleterious mutation characteristic of hereditary disorders such as sickle cell trait, beta thalassemia, infant phenylketonuria, and Huntington disease. They can also be used as molecular biological fingerprints to help trace the source of a sample of DNA to a specific individual. RFLP suffers from several important limitations, the most critical of which was the requirement of the relatively high quantities of DNA to employ as substrate for these restriction enzymes. Today, a dramatically more sensitive set of molecular diagnostic tools based on polymerase chain reaction PCR have largely supplanted RFLP analysis. Medical application of the polymerase chain reaction. As described in chapter 39, the polymerase chain reaction PCR employs a thermostable DNA polymerase and appropriate oligonucleotide primers to produce thousands of copies of defined segment of DNA from a minute quantity of starting material. PCR enables medical, biological, and forensic scientists to detect and characterize our DNA present initially at levels too low for direct detection. In addition to screening for genetic mutation, PCR can be used to detect pathogens and parasites such as Trypanosoma cruzi, the causative agent of Chagas disease, and Neisseria meningitidis, the causative agent of bacterial meningitis, through the selective amplification of their DNA. Recombinant DNA provides an important tool for studying enzymes. Highly purified samples of enzymes are essential for the study of their structure and function. The isolation of an individual enzyme, particularly one present at low concentration from among the thousands of proteins present in a cell, can be extremely difficult. By cloning the gene for the enzyme of interest, it is generally possible to produce large quantities of its encoded protein in E. coli or yeast. However, not all animal protein can be expressed in their appropriately folded, functionally competent form in microbial cells, as these organisms cannot perform certain post-translational processes taking specific to high organisms. In these instances, options include expression of recombinant gene in cultured animal cell systems or by employing the baculovirus expression or vector of cultured insect cells. For more details, see later chapters. Recombinant fusion proteins are purified by affinity chromatography. Recombinant DNA technology can also be used to generate proteins specifically modified to render them readily purified by affinity chromatography. The gene of interest is linked to an additional oligonucleotide sequence that encodes a carboxyl or amino terminal extension to the protein of interest. The resulting fusion protein contains a new domain tailored to interact with an appropriately modified affinity support. One popular approach is to attach an oligonucleotide that encodes six consecutive histidine residues expressed. His tag protein binds to chromatographic supports that contain an immobilized divalent metal ion such as Ni2 plus or Cd2 plus. This approach exploits the ability of these divalent cations to bind his residues. Once bound, contaminating proteins are washed off and his tagged enzyme is eluded with buffers containing high concentration of free histidine or imidazole, which compete with the polyhistidine tails for binding to the immobilized metal ions. Alternatively, the substrate binding domain of glutathione S transferase GST can serve as a GST tag. This figure illustrates the purification of GST fusion protein using affinity support containing bound glutathione. See, here is GST-T and here is the enzyme. This GST-T contains plasmid encoding GST and here is the cloned DNA encoding enzyme. They are ligated together and transfected into cells, added inducing agent and then break the cells. Applied to glutathione GSH affinity column and the cephros bead then separate and then elute with GSH, treat with thrombin. The GSH, GST complex and the enzyme are separated. This is the use of glutathione S transferase fusion to proteins to purify recombinant proteins. Now, the addition of an N-terminal fusion domain may also help induce proper folding of the remainder of the recombinant polypeptide. Most fusion domains also possess a cleavage site for a highly specific protease, such as thrombin, in the region that links the two portions of the protein to permit its eventual removal. Site directed mutagenesis provides mechanistic insights. 1. Once the ability to express a protein from its clone gene has been established, it is possible to employ site directed mutagenesis to change specific amino acid residues by altering their codons. Used in combination with kinetic analysis and X ray crystallography, this approach facilitates identification of the specific roles of given amino acid residues in substrates binding and catalysis. For example, the inference that a particular amino acid residue functions as a general acid can be tested by replacing it with amino acid residue incapable of donating a proton. Ribozymes, artifacts from the RNA world. Czech discovered the first catalytic RNA molecule. For many years after their discovery, it was assumed that all enzymes were proteins. However, while examining the processing of ribosomal RNA, rRNA molecules in the ciliated protozoan tetrahymena in early 1980s, Thomas Czech and his co-workers observed that processing of 26S RNA proceeded smoothly in vitro even in total absence of protein. They subsequently traced the source of this splicing activity to a 413 base pair catalytic segment of the RNA, which they termed as ribozyme, the discovery that earned Czech a Nobel Prize. Several other ribozymes have since been discovered. The vast majority catalyze nucleophilic displacement reactions that target the phosphodiester bonds of the RNA backbone. In small self-cleaving RNAs such as hammerhead or hepatitis delta virus RNA, the attacking nucleophile is water and the result is hydrolysis. For the large group 1 intron ribozymes, the attacking nucleophile is 3-hydroxyl of the terminal ribose of another segment of RNA and the result is a splicing reaction. The ribosome, the ultimate ribozyme. The ribosome was the first example of a molecular machine to be recognized, a massive complex comprised of scores of protein subunits and several large ribosomal RNA molecules. The ribosome performs the vitally important and highly complex processes of synthesizing long polypeptide chains following the instructions encoded in a messenger RNA mRNA molecule. For many years, it was assumed that rRNAs plays played a passive structural role or perhaps assisted in the recognition of cognate mRNAs through a base pair pairing mechanism. It was thus somewhat surprising when it was discovered that RNAs were both necessary and sufficient for catalyzing peptide synthesis. The RNA world hypothesis. The discovery of ribozymes has had profound influence on the evolutionary theory. For many years, scientists had hypothesized that the first biological catalysts were formed when amino acids contained within the primordial soup coalesced to form simple proteins. With the realization that RNA could now both carry information and catalyze chemical reactions, a new RNA world hypothesis emerged in which RNA constituted the first biological macromolecule. Eventually, a more chemically stable oligonucleotide DNA superseded RNA for long-term information storage, while protein, by virtue of their greater chemical functional group and conformational diversity, dominated catalysis. If one assumes that some sort of RNA-protein hybrid was formed as an intermediate in the transition from ribonucleotide to polypeptide catalyst, one needs to look no further than the ribosome to find the presumed missing link. Why did not protein take over all the catalytic function? Presumably, in the case of ribosome, the process was both too complex and too essential to permit much opportunity for possible competitors to gain a foothold. In the case of small self-cleaving RNAs and self-splicing introns, they may represent one of the few cases in which RNA autocatalysis is more efficient than development of a new protein catalyst. So, here we are completing the chapter. Let's study the summary. Enzymes are efficient catalysts whose stringent specificity extends to kind of reaction catalyzed and typically to stereochemistry of their substrates. In many enzymes, the suite of chemical tools available to facilitate catalysis has been expanded using a non-amino acid molecule. When tightly bound, these inorganic and organic molecules are referred to as prosthetic groups. If loosely bound, dissociable, they are referred to as cofactors. Coenzymes, many of which are derivatives of B vitamins, serve as shuttles for commonly used groups such as amines, electrons, and acetyl groups. During catalysis, enzymes redirect the conformational changes induced by substrate binding to effect complementary changes that facilitate transformation into product. Mechanistic strategies employed by enzymes to facilitate catalysis include the introduction of strain, approximation of reactants, acid-base catalysis, and covalent catalysis. Amino acid residues that participate in catalysis are highly conserved through the evolution of enzymes. The ability to change residues suspected of being important in catalysis or substrate binding by site-detected mutagenesis provides important insights into the mechanism of enzyme action. The catalytic activity of enzyme reveals their presence, facilitates their detection, and provides the basis for enzyme-linked immunoassays. Many enzymes can be assayed spectrophotometrically by coupling them to NADPH-dependent dehydrogenase. Assay of plasma protein includes